Welcome to This Explainer. Today, we're exploring a massive paradigm shift in how we understand our very own blueprints. For the longest time, the rules of genetics were treated as this master code, literally written in stone. But as we'll uncover today, the genome is actually a highly dynamic, incredibly environmentally responsive system. It's way less like a printed instruction manual and more like, well, a vast interactive switchboard. Okay, let's dive into this. Here is our roadmap for today. We're going to start with the central dogma. Then we'll look at the power of micromutations, the gradualism debate, modular genes, the myth of junk DNA, and finally wrap it up with epigenetics. I promise you, this journey from basic DNA structure all the way to modern epigenetics is one incredible interconnected puzzle. All right, section 1, the central dogma, our classical genetics. To really grasp this paradigm shift, we first have to understand the baseline rules of molecular genetics that an entire generation of scientists was taught. This all starts with Francis Crick's famous framework. The classic, canonical flow of information in life is actually wonderfully simple. DNA is the ultimate boss. It holds the information, and it passes those instructions down to an intermediate messenger called RNA. That RNA is then read out to build proteins. The major implication here? DNA is the absolute rule giver. It knows what's happening, and the flow of information goes strictly in one direction. Now, why are these proteins so absolutely crucial? Well, because proteins are the workhorses of the body. They physically hold the shapes of our cells together, they act as messengers like hormones, and they form the enzymes that spark chemical reactions. But here's the key. Their function relies entirely on their three-dimensional shape. See, proteins are made up of amino acids that are either attracted to or repelled by water, which forces the whole protein to fold into a very specific shape. That shape then fits into receptors in the body perfectly. It's exactly like a lock and key. Moving right along to section 2, the power of micromutations, essentially the power of typos. So what happens when that perfect lock and key system makes a microscopic mistake? Well, a micromutation happens when just one single letter, literally one nucleotide, in the DNA sequence gets accidentally miscopied or changed by something in the environment, like radiation. Now, because there are 64 possible combinations of DNA letters coding for just 20 amino acids, there's a ton of redundancy built right in. That means some of these single letter changes are totally neutral. They don't change the amino acid at all. But other times, they can radically alter the protein's final shape. Which leads us to a massive question. What actually happens to a human being when just one single letter in this vast genetic code changes and a protein's shape is altered? Well, it really depends on the type of typo. There are 3 classical types of mutations. A point mutation just swaps one letter for another, which might just slightly tweak a protein's efficiency. Not always a huge deal. But a deletion or an insertion, that actually removes or adds a letter. And that causes a massive frame shift, completely scrambling all the genetic meaning downstream and usually destroying the protein's function entirely. And this brilliantly illustrates the real-world human impact of those tiny typos. Take PKU, for example. A single mutation breaks an enzyme, which leads to a toxic buildup that destroys the nervous system. Or TFM, where a broken androgen receptor means a person with male XY chromosomes is born phenotypically female, simply because their body cannot respond to testosterone. Or there are these fascinating cases in the Dominican Republic where an enzyme that makes testosterone works incredibly slowly. Because of that, individuals are born appearing female only to transition to male at puberty when a surge of hormones finally crosses the threshold. A single shape change in a protein can dictate entirely different life outcomes and gender phenotypes. Crazy, right? Section 3. The gradualism debate, slow versus rapid evolution. So these tiny, incremental mutations didn't just explain diseases. They fueled a massive theoretical and even politically charged clash over how evolution actually happens. We ended up with 2 deeply contested worldviews. On one side, we had the gradualists. These were mostly sociobiologists and evolutionary biologists. They argued that evolution is driven by constant, slow, incremental changes. 1% genetic advantage here, a tiny mutation there, all driven by constant competition. But on the flip side were paleontologists like Stephen Jay Gould, who proposed punctuated equilibrium. They argue that evolution isn't gradual at all. Instead, literally nothing happens for millions of years, and then boom, rapid bursts of evolutionary change. This was an intensely heated clash between biologists studying modern gene copies and geologists looking at ancient rocks. The paleontologists looked at incredibly detailed fossil records, like timelines of Caribbean snail shells, and they saw exactly what their theory proposed: massive gaps, and rapid jumps that totally contradicted the slow, step-by-step gradualist model. Of course, the biologists fired right back. They argued that studying fossilized snail shells couldn't possibly tell you how something complex, like a brain, evolves. The gradualists challenged the paleontologists to prove how rapid bursts of change could even be physically possible at a molecular level. Which takes us right into Section 4, modular genes and splicing. Enter the splicing enzymes. Ironically enough, it was modern molecular biology itself that started supplying the evidence that shattered the old gradualist rules. Under the old central dogma, the rule was simple: one gene equals one protein. A gene was thought to be this continuous, unbroken stretch of DNA. But scientists discovered that genes are actually modular. They're broken up into coding chunks, called exons, which are separated by long stretches of non-coding space called introns. To make a protein, special proteins called splicing enzymes act like molecular scissors to just cut out the introns and paste the exons together. And because of those molecular scissors, one single gene sequence could theoretically generate 7 or more entirely different proteins. Depending on the tissue or the specific context, those splicing enzymes can mix and match different combinations of exons. So the whole one gene, one protein rule, officially dead. The genome was vastly more complex and combinatorial than anyone ever realized. Section 5, the myth of junk DNA. Let's look at the vast emptiness. Now, what's really interesting here is how this leads us right into the next massive contradiction to the central dogma, which involves the vast emptiness of the genome itself. When scientists first realized that genes were separated by huge stretches of DNA that didn't code for proteins, they literally called it junk DNA. But think about the sheer scale of this. 95% of our DNA is non-coding. Well, it turns out this isn't junk at all. That 95% is the crucial instruction booklet. It contains the on and off switches, the promoters and repressors that actually tell the 5% of coding genes when to activate. And this totally flips the central dogma on its head. DNA doesn't act like the boss calling the shots. Actually, DNA knows squat on its own. It's just the readout. It requires messengers called transcription factors to come in and flip those genetic switches. And what triggers those transcription factors? The environment. It could be low cellular energy, a surge in testosterone, or even a rat smelling her newborn baby. An environmental signal activates a transcription factor, which then binds to a promoter and turns an entire gene network on. Finally, Section 6, the power of epigenetics, shaping your genetic destiny. So, if the environment can turn genes on and off, how deep does that influence actually go? Well, we know the environment can trigger transcription factors to access our genes, but the DNA itself is physically wrapped in structural proteins called chromatin. Epigenetics looks at how the environment can physically alter our access to that DNA permanently. Through mechanisms like chromatin modeling and methylation, early experiences can permanently silence a gene. For example, research shows that how a rat mother licks and grooms her baby can cause permanent, lifelong structural changes to the baby rat's DNA. It literally locks away or opens up stress hormone genes for the rest of its entire life. So, the crucial point is captured beautifully in this quote from the source material. Fertilization is all about genetics. Development is all about epigenetics. The actual DNA sequence you are born with, your genetics, is really just the starting point. The context, the environment, all those if-then clauses built into your 95% non-coding DNA, that is what actually sculpts development. Which leaves us with this final, truly provocative thought. We started by looking at genes as a rigid rulebook, right? A master code dictating our fate. But if our environment, from the cellular level all the way up to our childhood experiences, can physically and permanently shape our genetic destiny, how much of you is actually written in stone? It's a fascinating question to ponder. Thank you so much for joining me for this explainer, and keep questioning the incredible biology that makes you, you.